Le temps qu'il rentre, voilà.
C'est ouvert, j'ouvre la salle d'attente.
Et pourquoi pas que on le fait et Henri peut dire change.
Ah, mais ça change rien. Ça changera rien. Ok.
C'est le logiciel qui va pas.
Ok. Tu veux dire qu'il nous l'envoie à nous et que nous on change les slides?
Oui.
Okay, welcome everyone to this SEM Friday seminar. We're very happy to have you, and we're very, very sorry to had you waiting for some time. But we're experiencing a couple of technical problems here, actually for the first time since we're doing this via Zoom. So there's a first time for everything. And again, we're very, very sorry for that. We're trying to solve this as soon as possible to get a nicer picture to you all. So we'll probably start in a couple of minutes.
Henrik, tu peux envoyer ton PDF à moi ou à Ali.
Ouais,
et on va partager nous l'écran et je pense que ça marchera mieux sur Adobe.
Comment je vais mettre quelqu'un habitué, c'est webmail.
T'envoies un mail.
Pour joindre.
Ouais, pour rejoindre.
Pour joindre.
Option.
Là, il est où?
Okay, sorry everyone. I use Linux and it's always difficult. Yeah, okay, desktop.
No, that's not the point. It's always difficult because some people seem to be using Windows, right? If everyone was using Linux, everything would be so easy.
It does work on Linux, though. It should work normally.
Okay, Alison has my presentation now. So I could, there's no many presentations, so I can always tell Alison next slide or something like that.
Yeah.
Yes, that would be the
So
best.
then I can do the presentation. You don't see me on screen while I talk, and then
we will see you. We will see you.
Yeah.
Leave your camera on. You'll be on the corner, I guess.
Yes. Okay. Not my best day, but it's fine.
This is happening the day where we even have guests from Switzerland.
Yes.
How embarrassing.
50 people in the room.
Very sorry again.
Is there 50 people already?
Yes.
And
we have in CBGP and there's like 10 here. So.
Does that work?
Oh, and then she's using Mac.
Wrong now?
This won't work either.
No?
Why? Maybe it's because we are sharing with our screen here. Huh?
I had the same issue when I did my SDF.
Ah, yeah?
You know how to do the full screen, right, Ali?
Yeah.
So maybe as long as we're trying to fix new problems, again, I'm very happy that everyone is here. The SEM is brought to you by the LabXMEB, and we're very thankful to Agrinium for supporting us with this Zoom webinar license. And without further ado, Ali, you can start introducing.
Hello, so yeah, we're very pleased that Henrik Frago has agreed to do the seminar today at quite short notice, which is great. So Frago did his PhD at the University of Valencia, looking at population dynamics in moths. Then
yes.
On to white moths. Then he moved on to a postdoc at Oxford, and then a Marie Curie postdoc at Wageningen in Holland, where he was looking at interactions between herbivores, their host plants, their symbionts, and natural predators, looking at related questions at how symbiont-mediated protection in mites and how symbiont-mediated protection acts via plant volatiles. 4 or 5 years ago, Frago got a permanent position at CIRAD, and he's now looking at related questions with thrips and whiteflies. So thank you very much, Frago.
Yeah. Okay. So can you hear me?
Yes.
Yeah. All good. Can I start?
Sure. Go ahead.
Yes.
Okay.
Maybe before you start, so if people have questions, you can use the Q&A tab that you will find at the bottom of your screen. to ask your questions already during the talk, and we'll
Okay.
answer them afterwards.
Okay, yeah, good. So, perfect. Yeah, so thank you very much, Alison, for the presentation. As you have my full screen, when I get into next slide, I will tell you like something like next. Okay. So yeah, as Alison said, my name is Enric Frago, and I will present you today a work that we did in collaboration with Sharon Zitinska from the University of Liverpool, which is a meta-analysis on the benefits and costs of hosting secondary endosymbionts in sap-sucking insects. This work was done by Sharon and myself, and also by Karim Tihiwart, who is a student doing the PhD with me at this moment. And Karim and myself, we are both at CIRAD, which is the French research center for research in agriculture in tropical countries. This project, there's a mouse moving around. Maybe Alison, stop moving your mouse. If not, it's a bit disturbing. Sorry. So this project is the product of a cost action, which was a 5-year project from the European Union. Cost actions are projects that fund networks of scientists around Europe. In our cost action was about insect-plant microbe interactions, and Sharon and myself were in charge of the microbes living inside the insects, which is this meta-analysis. Next. Yeah, so we are now more and more aware of the importance of microbiota for all animals. We know now that we cannot understand the biology of animals without understanding the microbes that live inside. For example, in humans, we have our guts full of and skin full of microbes, and there's quite some recent studies, but also quite some older ones. For example, this one about microbiome and obesity, where the researchers selected couples of twins, one with the problem of obesity, the other one without, and by transferring the microbiota of these two persons into a mice, they managed to transfer this obesity problem, which shows that the obesity problem was not the genetic background of the person because they were twins, but the microbiota. So in the future, do not get surprised if your doctor gives you a poo pill, which is a pill with the healthy microbiota of a healthy person that can help you fight diseases like, for example, the Clostridium difficile variants that are resistant to antibiotics. Our skin smells, and this can attract mosquitoes that can transmit lethal diseases, and this smell can be also mediated by the microbes in our skin. And even sexual communication in hyenas has been found to be mediated by microbes. Next. But let's move on and talk about insects now. So insect symbionts can be bacteria, fungi, and protozoans. I will mostly talk about bacteria because it's the group that we have done more work, but there's some recent exciting evidences about fungi and protozoans. So this symbiotic association with hosts, they have been happening in all known taxa, because they are quite important sources of phenotypic innovation. For example, the symbionts can allow an insect to fit on a new host plant, and this can open a new niche for this insect. Or, for example, these symbionts can protect their hosts from natural enemies, so these insects they can colonize new habitats that were before too dangerous for them. So they can be the source of diversification. Together with Shannon Zitinska and also Nina Faturus in Wageningen, we recently published a book chapter. I take the advantage to invite you to read it. So where we did a revision on how symbionts affect the biology of herbivorous insects. In the last 5 years, there's been like tons of reviews on insect symbiosis, and when we wanted to write this review, we didn't know what to do because there was already too many reviews. So we decided to do an order by order revision. So we went each order and we talk about these orders and what's known in their symbionts. So we hope it's a reference that can be used for people that says, I don't care too much about insects, but I want to know what's inside dipterans, and then you go to the specific section of the chapter. So, for example, in hemipterans, you can see that I marked it with a red arrow, which is the group I will talk about. We know that the symbionts can provide nutrition, they can help in degrading the cell walls in plants, but they can also suppress plant defenses, and they can also play important roles in defense against natural enemies. Next. Thank you, Alison. Today I will talk about phloem feeders, which are mostly hemipterans and commonly known as aphids, whiteflies, sylleats, and mealybugs. Why this group of insects? Well, this group of insects, they have acquired obligatory symbionts, all of them, and each big group of insects have acquired a particular symbiont. So we could not understand the biology of these groups today without their symbionts. For example, all aphids carry Bugnera, and these symbionts provide their insects with essential amino acids that the insect cannot acquire from the diet. So, as I've said, when these groups evolved and acquired these symbionts, this is when they diversified in our planet. So, on top, sorry. So these symbionts, as I've said, they provide essential nutrients. They are inside bacteriomes, which are kind of organs, where the bacteria are well packed inside the host. But on top of these obligatory symbionts, facultative these phloem feeders also carry facultative endosymbionts that can be found in bacteriomes or not. But interestingly, they provide conditional benefits. They provide benefits in some situations, and they are not always present. They are not always in all populations of a species. This is why we are interested in them in this meta-analysis, because doing a meta-analysis on obligatory symbionts would be quite stupid, because the fitness effects of losing the symbiont is always the same. The insect dies. But in endosymbionts, we can try to evaluate the costs and the benefits of these symbionts. Next. So here an example that I always present when I talk about these symbionts is the Asian citrus sylleat. You can see on the top left picture, there's the whole insect. In the abdomen, you see a yellow organ, which is not an organ, is a microbiome, is a package of symbionts that if we cut it sagittally, we can see on the right side, we can see that this bacterium has two different layers, one external, which is Carsonella, that provides essential amino acids, and one internal, which is Protella, which produces toxins, and these toxins are thought to make the insect toxic and protect it against natural enemies. Next. So in whiteflies too, we know there's quite some facultative symbionts like Hamiltonella, which is depicted in green, or Arsenophonus in yellow. If you take a look at the bottom right picture, you can see a whitefly, and you can see in the abdomen a yellow spot, which is the bacterium. So this is to show you that these symbionts are not somewhere in the insect. They are located in very specific parts of the insect. They are transmitted from mother to offspring because the symbiont colonizes the embryos, and they are vertically transmitted. So the evolutionary destiny of the host and the bacteria are very tightly connected. Next one. So if we take a look at aphids, this is another paper by Sharon and his collaborator, who explored how many insect species were carrying facultative symbionts. So you can see on the left side of the slide all these different symbionts that we know facultative. The obligatory is Bugnera, and is always present. Of these facultative symbionts, and you can see in the graph in yellow how many species carry a given symbiont, and in red in yellow how many species do not carry the symbiont. And in red how many species do carry the symbiont. And you see that, for example, Wolbachia is present in almost half the population, half the species of Aphids studied, whereas Hamiltonellas is only in 1/3. But then it's important to know, again, even if one species carries Hamiltonella, not all populations of the species will carry the symbiont. Next. So if we go a bit more in detail of of the P-aphid example. So aphids are a good model system for symbiosis because they are easy to culture, easy to manipulate, and the P-aphid is probably the aphid model for symbiosis. As all aphid, it carries Bugnera, but it also carries several different facultative symbionts. Of course, they are not all symbionts are found in the same individual. Usually, P-aphids carry no facultative symbionts. One symbiont is maybe the most probable situation. 2 is quite normal. More than 2 is possible, but quite rare. But among these 7 symbionts, we can have, for example, Regiela insecticola, which provides protection against fungal pathogens, Hamiltonella defensa that protects against parasitoids, and Serratia symbiotica that protects against heat shocks. You can see in the picture on the right side of the slide, you can see a parasitic wasp that is laying an egg inside a little baby aphid, and this egg will develop into a larvae that will eventually kill the aphid. But if this aphid carries some strains of Hamiltonella, the aphid will be immune to this. The larvae will never develop into adult. Next. So I've talked about 3 different groups of phloem-feeding insects, but there's also an important group of phloem-feeding insects which are Heterotrons, including stingbugs, shieldbugs, and truebugs. In these groups, the associations with symbionts are a bit less intimate. The symbionts are not found inside the body, but in the digestive tract. And if I've said that in aphids and whiteflies, the transmission is via the embryo, in these groups, vertical transmission is a bit more complicated. For example, in Megacopta punctatissima and Megacopta crivaria, the mother, when the mother lays the eggs, it deposits a group of symbiotic particles. You can see them in the picture in the left side. You can see in white the eggs, in yellow the babies, and in black the symbiont capsules. So when the offspring gets out, this offspring needs to pick up the symbionts to acquire them. So this is much less reliable than transmitting it through the embryo. If I was a mother and I want to ensure that my son has the symbiont, I would prefer to transfer it through the egg in the embryo than doing this weird thing. But this is why these associations are less intimate. For example, in the other side of the slide, you can see Rhiptopterus pedestris, which is another stingbug. You can see in the bottom figure, you see in blue the gut of the insect, and in green is the symbiont. So it's found, even if the association is not as intimate as in aphids, for example, you can see that there's the gut of the insect is quite well colonized by this symbiont. Next. Another interesting thing with phloem-feeding insects is that we can manipulate symbionts. We can collect these insects in the field, assess which symbionts they carry. For example, in aphids, we can even establish clonal lines because aphids can be kept reproducing asexually in the lab. And these lines can be cured from facultative symbionts using antibiotics. In aphids, we need to use antibiotics that kill the facultative but not the primary. If not, if we kill the primary, we kill the aphid too. In aphids, we can even infect aphids with new symbionts via microinjection. We can take the symbiont from one species and transfer it to another one. In whiteflies, curing symbionts is difficult with antibiotics, so many studies have used introgression of symbionts via crossing. Basically, we have 2 lines, one with symbiont, the other without, and we transfer the symbiont via mating these 2 different lines, and then by back crossing several times, we try to homogenize the genetic background of the whiteflies so that we have one line with a symbiont and the other one without. Next. So, yeah, the overarching question of our study was that we know that these facultative symbionts provide benefits against abiotic stressors. They can help them use new plants, they can protect them from pathogens, they can even change the color of the aphids, they can protect them against natural enemies. So if they are so useful, why they are not always there? So this is because they are often costly, and these costs and benefits makes that carrying them is not always useful. And this is why we wanted to do this, one of the reasons why we wanted to do this meta-analysis. Next. So the three main questions I'll try to answer is which groups have been sufficiently experimentally examined for the effects of symbionts on the host. The second one is whether there is a trade-off between costs and benefits of symbionts. And the third one is whether these symbiont effects are species dependent. Next. So I don't know which, it's my first webinar, so I don't know the type of audience that is on the other side of the cable. So I don't know if you are all familiar with meta-analysis. So I will spend like a few slides trying to explain a little bit how meta-analysis works and how we did that. Sorry. So a meta-analysis is basically a statistical analysis that combines the results of multiple scientific studies. So we started by looking for specific keywords in the Web of Science. Next. For example, for the person passing the slides, now there's quite some next, but then it's over. So don't worry too much. So for the keywords in the Web of Science, for the whiteflies, for example, we add always symbiont by the end, and then whiteflies and common names as a common name, and then the most common genus that have been studied for whiteflies. And then, based on this search, we established several selection criteria. Next. Which in our case was that these studies needed to experimentally test for symbiont effects using antibiotics, introgression, and microinjections, as I've explained before. As in all meta-analyses, we need to obtain data and to fit our statistical models with this data, and we need that the authors of the paper reported the mean value, the standard error of this value, and the number of replicates, because these are the data that we use to fit the statistical analysis. Next. Then, that's the most painful part, which is extracting these mean values and these standard errors, which we did with a free program called WebPlot Digitalizer, thanks to the developers, by the way. So we basically extracted these graphs from the papers and we measured the mean and the standard errors. Sometimes the data is in the text, so then it's very easy, you just copy paste, but sometimes you have to extract it from this WebPlot Digitalizer. Next. So we were interested in few life history traits, including body size, development time, lifespan, fecundity, and survival and parasitism. This is the life history traits that we used to infer if carrying symbionts or not was good for the insect or not. Then we calculated standardized measures or effect sizes using the HGSD value, which is a classic statistic classic value in meta-analysis. And basically this effect size is telling us from each data set, from each paper, if we have a positive effect size, it means that the symbiont was doing something good to the insect. If the value was negative, it means that the effect was negative or a cost. And then we basically fit the program similarly to a GLM model, and then we validated this model to assess if there was publication bias or p-hacking. P-hacking is when authors only publish when the p-values are lower than 0.05. Next. So let's go to the results now. To answer the first question, which groups have been sufficiently experimentally examined for the effects of symbionts on the hosts? So you can see here a table where I represented the three groups we managed to get enough data, the Heterotrons, the whiteflies, and Aphids. The first column is the number of papers we obtained from the Web of Science using our keywords, so like more than 1,000 papers. But some of these papers, just reading the abstract, you already knew that they were not interesting because they were, for example, natural observations of symbionts in the wild without experimentation. So these papers were already discarded. So after our criteria, we only end up with 13 papers from Heterotrons, 8 whiteflies, and 68 for Aphids. And then the third column is the number of data points. Of course, there's more data points than papers because some papers were measuring different life history traits or even presenting data from different insect species. Okay, and finally, you can see in the last column the number of species: 14 species of Heterotrons, 13 species of Aphids, 1 single species of whitefly. Quite disappointing. And for Aphids, there's 13 species of Aphids in this meta-analysis, but 90% of the data comes from a couple of species, the pea aphid and the black bean aphid. So as phloem feeders, there's also sylleats and mealybugs. No, only a couple of papers on sylleats, none of mealybugs, so we couldn't do any meta-analysis on that. We don't know why, maybe because no one has got enough interest on these groups, or maybe because curing these symbionts experimentally in these groups is very difficult, so no one has published these problems of curing the insects. As I've said, quite some diversity in terms of Heteropteran species, only Bemisia tabasi in terms of the whiteflies. And Bemisia tabasi is a very important pest with quite some different biotypes, so some of them can be even considered as different species, but still quite related phylogenetically speaking. And in aphids, quite good data, but limited to a couple of aphids. And sometimes in one of the aphids, the black bean aphid, for example, almost all data comes from a single laboratory in Switzerland. So what we can infer is quite limited sometimes. Next. So this is to answer the second question. No, it's still the first question. I wanted to just show you this circular plot, which shows the whole data set. What we represent here is like the zero in the whiteflies, which are in yellow. You can see the y-axis, which is the effect size. You can see that the 0 is represented by this continuous line. Okay, so every point here is a data set that a data point that we extracted from the papers. And if the values are below 0, it means that the symbionts are costly. If they are above, it means they are beneficial. So we can see, for example, that for whiteflies, we have much less data than for aphids. That's another useful thing of this type of representation. And we can also see on the legend, nicely colored, that the different life history traits are represented in different colors. So we can see the traits that we have explored in this study. If we check, for example, aphids, we see that there's quite some negative values which are below 0, below this continuous line, and we have a lot of points which are in green. These are costs in terms of fecundity. But if we go to the other side, we go up, 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 and we get above 0, we see a lot of purple points, which are survival to parasitism. This is the benefit that the aphids can get from the symbionts, from protection against natural enemies, for example. I'll go into more details after that by summarizing the data. It's just to give you a general overview. Maybe the most interesting thing from this representation is that in Heteropterans, we have maybe 10, 15 negative values, and then after that, we have an explosion of positive effects and of very large effect sizes. This suggests that in many cases, these symbionts in Heteropterans are super beneficial, are so beneficial that we think that most of these, in most of these species, the symbionts are in fact obligatory. We don't think that most of the insects would survive in nature without the symbiont. We've got quite some cases in which mortality was larger than 50% when the symbiont was removed. These insects, you can keep them in the lab, but I'm not sure they would survive in nature. Next. So this is like probably more clear and more synthetic, where we have summarized the data from the circular plot for the different study groups. You can see in the horizontal line the effect sizes for each of the different groups, for Heteropterans, for whiteflies, and for aphids. And in the y-axis, you see the different life history traits that we have studied. As I've said before, for Heteropterans, if you take a look at the scale of the effect size for Heteropterans, the scale goes from 0 to 6, whereas for the others, for aphids and whiteflies, it never goes higher than 1. So effect sizes in Heterotrons are huge. Losing these symbionts imposes strong cost, so carrying them is really, really beneficial. So that's what is represented here. An important note is about development time. We transform development time as the inverse value. So a positive effect, a beneficial effect, we have considered a beneficial effect as a shorter development time, because usually insects that develop faster, they can escape natural enemies. They can more easily escape from natural enemies or escape from adverse abiotic conditions. So we considered that short development times is a good thing. And for that, we just transform the effect sizes by multiplying by -1. Then for whiteflies, for Bemisia tabasi, because it all comes from a single species, we can see that there's a clear positive effect for fecundity and for development time, but apparently there's no associated costs to carrying these symbionts. It's only beneficial. What we think it's possible in this situation is that these facultative symbionts are in fact kind of assisting nutrition in the insect. As it has been found in some aphids recently, some facultative symbionts can complement the obligatory symbiont in acquisition of amino acids from the diet. So we think that it's possible that this is what's happening in Bemisia tabasi, that these symbionts are in fact only having a positive effect because they are kind of nutritional or helping the insect to deal with the phloem sap, which is quite poor in nutrients. Then if we take a look at the aphids, the third column, then we see in agreement with our hypothesis, there's a trade-off between fecundity and resistance to parasitic wasps. So you can see that when these aphids carry these facultative symbionts, they produce less offspring. And this is quite bad, carrying a bacteria that it makes you less fertile or less fecund. But then you get a benefit in terms of resistance against natural enemies. If I was an aphid, I would prefer to be less fecund, but when a natural enemy comes around, I am protected, because natural enemies, specifically parasitic wasps, are very efficient natural enemies of aphids. This is why they are very commonly used in biocontrol programs. A single female wasp can attack like 100 aphids in a colony and completely eliminate a colony from a plant. So we think that this is probably the most interesting result of the meta-analysis, that even if there are some papers that have studied in specific situations this trade-off, we can now demonstrate that this, by considering all the data that we have for aphids, this trade-off is present. Next. So, just to tackle the third question on whether symbiont effects are species dependent, we could only analyze this for aphids because it's the only group we had enough data. And the answer is that yes, the effect is species dependent. And it's quite interesting also because if you take a look at the bottom part of the graph in parasitism, you will see that there's 3 aphids here: AP, which is the P aphid; AF, which is the black bean aphid. The P aphid is the picture bottom left. The next one is the black bean aphid. Then it's Cytobium aveni, which is the grain aphid, SA. So this aphid is an important pest too, but its symbionts do not protect it against parasitic wasps, as you can see in the bottom part of the graph. And if you take a look at the effects on fecundity, it's quite interesting that Cytobium aveni, the SA, is not paying any cost of carrying the symbionts. On the other hand, AF and AP, the P aphid and the black bean aphid, they are paying a cost in terms of fecundity for carrying the symbionts. So again, we can see that this trade-off in terms of defense and fitness is species dependent. Overall, we found an effect, but if we go into the details of the P aphid, we see that P aphids are protected from natural enemies, but they pay a cost, black bean aphids too, and the Cytobium aveni no, at least with the studies that have been conducted so far. Next. So now there's another table here which shows basically the same, the effect size for lifespan, fecundity, and parasitism for the different symbiont species. Not much happening here. If we take a look at the bottom part again, in terms of parasitism, we see clearly Hamiltonella defensa is defensive. This is the first symbiont that has been found to be defensive, and there has been lots of studies on this symbiont. So our meta-analysis tells us what we already knew, that Hamiltonella is a defensive symbiont. Rickettsia is also defensive, but what is a bit surprising is that when we take a look at the costs in terms of fecundity, only Rickettsia is costly, but not Hamiltonella defensa. Probably this is because there's some Hamiltonellas which are not very defensive, so maybe they are not very costly. But this is something that we cannot really say with a lot of. We don't have strong arguments to really test this across all the different studies that we put in the meta-analysis. Next. So, to summarize, which groups have been sufficiently experimentally examined for the effects of symbionts on the host? Enough data for aphids and heteropterans. We only have one species of whitefly, so we need more studies on different whitefly species. We don't have any data for psyllids and mealybugs. For the second question on whether there is a trade-off between costs and benefits, yes, in aphids, and mostly driven by Hamiltonella, impy, and black bean aphids. But there's no trade-off in whiteflies and heteropterans, as we only found beneficial effects. And we think that in heteropterans, the effects here are extremely positive on symbionts, so these symbionts are likely to be obligatory and not facultative. The third question is whether these symbionts effects are species dependent. We only have data for aphids, and most symbionts that are protective are also costly, but mostly in aphids and black bean aphids. And the most clearly defensive symbiont are Hamiltonella and Rickettsia. And here we know we lack a lot of data on multiple infections because, as I've said, carrying only one facultative symbiont is more the exception than the rule. And we need to know what happens when these symbionts are in communities with other symbionts. There are some papers that are considering these interactions, but not enough to be included in the meta-analysis. Next. Yeah, here I wanted to present a couple of slides on my current research because I'm not doing much work on symbiosis now. And I took the advantage to present another of Karim's project on his PhD project, which is on the experimental community ecology. We basically are now getting interested in making quite complex communities. Karim has done experiments with in mesocosm cages, microcosm cages, with replicating these communities and then manipulating the communities. For example, removing higher order predators like the Nesidiocoris volucer predator, or removing some herbivores. And then we wanted to know how this can affect suppression over the herbivores and plant health, but also because we did these experiments in the long term during several months, we wanted to test how these communities are more or less stable depending on the interactions at the natural enemy level. Next. And maybe one of my future dreams will be to include some symbionts in these systems. We already published a study a few years ago about how these symbionts can alter community dynamics, but we would like to do that in a more complex situation and see how this can alter bottom-up and top-down interactions in complex communities. Next and last. So that's all I wanted to say. Thank you very much for your attention. I don't know if you enjoyed it or you are just sleeping because I don't see anyone, but I hope you enjoyed it. Again, a big thank to Sharon Zitinska, who lead this project, and Karim Tijiwart, who is currently in CIRAD in La Reunion, where I was before. And of course, a big thank to the cost action, because this project is product of this meta-analysis, part of this large project. Thank you very much.
we even have people clapping, so I don't know where this is in your.
Yes, because I'm giving the talk in my institute, and there's like 10 people.
All right. So someone says great talk, so I guess people like the talk. There are some questions in the Q&R thing, so I can read them for you. Some Alexandra is asking how to distinguish obligate. Oh, you can read them too, but perhaps Alexandra can also turn on her microphone, or someone can turn it on for her if he wants to react. But I'll start asking the question. Oh, Alexandra? I can ask, so how to distinguish obligate from facultative symbionts? If you don't know it from the literature, is it experimentally? If it doesn't survive without a specific bacteria or any other clues, how do you define obligate versus non-obligate, basically?
Yeah. Well, the easiest thing to distinguish if a symbiont is obligatory is by removing it and then checking if the insect survives. So if the insect dies, then it's obligatory. If you want to know something about a specific species to know if the symbiont is obligatory or facultative, if it's an aphid, a whitefly, a psit, or a mealybug, I'm telling you that each one of them carries an obligatory symbiont that is obligatory, because when these species diverged, they acquired the symbiont, and then thanks to this acquisition, they managed to colonize new ecological niches. For stingbugs or other species, I guess you have to manipulate with antibiotics, or you can do a more genomic approach where you check if the symbionts provide essential amino acids, and then you can guess they are obligatory, but the experimental demonstration is always the best answer.
Just to follow up, so Alexandra, if you're happy with you can react if you want to say more. I'm wondering, is there such thing as an obligate symbiont for reproduction, like not obligate for survival, but you need them some way? Is there cases like this?
Well, I mean, I guess that if you don't reproduce, you don't survive.
Well, you could survive and not reproduce.
Yeah, but this is
still obligate, but.
Yeah, it's an evolutionary dead end. I mean, you can survive, but at the end, the species will.
That's why I'm asking if you are aware of any case like this.
Well, Wolbachia, if we think about sexual manipulators, Wolbachia is the clear example, but Wolbachia is mostly making many insects only producing females. So that Wolbachia is a kind of was considered as a selfish element that makes sure that the bacteria is transmitted always. More than this.
Well, okay, don't worry. It wasn't really in the question, so I was just.
Okay.
So there's a question by Besem who's asking, saying that in aphids the benefits are related to protection, and asking if there was any work that looked at the tipping point below which it's more advantageous to lose the symbiont, even in the presence of natural enemies. In other words, did someone look at the enemy's density below which having a protection is more costly than beneficial, basically?
Yeah. There is a paper, a Proceedings B paper by Christoph Borburger team. We always refer to the senior author because we remember the person, but it was done by someone else. I don't remember the name. Where they had a battery of P. aphids with different Hamiltonellas that had different levels of protection. Some of these Hamiltonellas were even non-protective. We know that there are some symbionts, some Hamiltonellas that they are there, but they don't protect against natural enemies. And they tested this hypothesis of whether more protective ones were more costly, and they found no correlation. So the story is more complicated than we think, because the symbiont can be super protective and not very costly, or not very protective and very costly. But these studies have been done in the lab against a single wasp species. And we know also from other studies that some symbionts can protect against some Hamiltonella strains can protect against some parasitoids, and some other strains protect against other parasitoids. So it's difficult to know in nature when carrying the symbiont is useful or not, because also natural enemy densities change a lot during the year. So I'm not sure I can answer the question very precisely.
But it seems, sorry, just to follow up, it seems that you could, from your computation of the cost and benefits, you could just, you know, even with a model, just assess at which frequency of encounter it becomes. Like if you never encounter the parasite, then you just have the cost base.
Yeah. I guess that what would be very interesting is to know the ecology of these aphids. Like if you live in an environment where there's less natural enemy pressure, then carrying a costly symbiont, expecting that at some point you will be attacked, would be quite not very useful. There's some work done in England too, in Oxford, where they tested that. They put aphids in different host plants, and these different host plants are known to carry different symbionts, and they explored whether in these plants there was more natural enemy pressure. The results were a bit mixed. So some of the results supported their hypothesis, saying that yes, there's more pressure of natural enemies in this plant, that there's more symbionts, but in some other plants it doesn't. So,
so Besam is here. So are you happy with the answer?
Yeah.
We can move on. Okay. Then Pradeep is asking. So Emmanuel, sorry, are you the one giving them the? Oh, okay.
Yes.
Pradeep, if you want, you can ask your question. If you turn on your microphone.
So
nice talk, actually. It's audible?
Yes, we can hear you.
Yeah, we hear you. Hello.
Okay, so it seems to have problems, but he's asking whether, yeah, we can hear you. Whether oleander aphid adapted to milkweed toxin is dependent on symbionts or independent of symbionts.
Yeah.
You know about this?
Hey, Pradeep, we cannot hear you very well, but I can read your question. So to be honest, I don't know. I guess that the usual procedure in these situations, I've seen some papers where they have explored these kind of questions, not with aphids scenario, where they have collected aphids from different plants. I don't remember the model system. It was not pea aphids, not black bean aphids. It was another species. I don't remember the name, but they collected aphids from 2 different host plants, and they found that in one of the host plants, there was always one facultative symbiont present. And when you see that, of course, you cannot do that only with 2 populations. You have to find the replicated populations in replicated plants in different landscapes and separated in space to avoid the spatial autocorrelation on these effects. If you always find that one of the symbionts is always present in one plant, then you can start wondering, maybe that's the effect. And then if you do genomics on this plant, on this symbiont, then you find that the symbiont has the capability of digest milkweed toxins, then you have a nice story. But yeah, first I would start by collecting the insects, maybe then curing them and trying to feed them on the plant where they are usually present and see if there's a fitness cost. Then you can start looking at the link between the symbiont and the plant.
Okay, so then Lucy is asking, saying great talk and asking, did you have in your meta study? Okay, so basically the question, I guess, is whether some of these facultative symbionts are in bacteriums and others in gut. I thought the bacterium was only for the obligate, but perhaps you can correct this.
Yeah.
If yes, any observation on gut symbionts and which one are more beneficial?
Yeah. Yeah, that's a tricky question also. So in aphids, whiteflies, mealybugs, usually symbionts are in bacteriums. Even if all these groups, they also carry bacteria in the guts, but not these highly evolved bacteria that provide essential amino acids. Then in the sting bugs, in the Heterotrons, most of these symbionts are in the guts. But in many situations, so one thing that I haven't said during the talk is that when we started analyzing the database for sting bugs, we had many cases in where the sting bugs, the Heterotrons, removal of the symbiont led to more than 70% of mortality. These studies were not included. It was our arbitrary decision. These studies were not included in the meta-analysis because we consider that more than 70% mortality is an obligatory symbiont. The symbionts are in the guts, sometimes in small crypts. So I would say that these are super intimate interactions that are almost as intimate as in aphids, maybe a bit less because these bacteria have lost less genes and so on, but they are super intimate. So in terms of gut symbionts, there's a nice gradient between very obligatory to quite facultative. But most of them are quite needed for the insect to have a decent fitness.
So Raquel is asking, what are the limitations of your studies, sir? And I would add to this, if I can, that you mentioned in the description that you have to deal with publication biases, p-hacking, etc.
Yeah.
But how do you account for this? And also, did you account for the error in this measurement that you use with the figures where you have to, extract the data? So I guess this sort of goes with the same.
Yeah.
So this is the technical side, and perhaps there are other limitations you have in mind which you can react.
Yeah. Well, for the most deep limitations that we have is what I've already mentioned. We were saying things about whiteflies based on a single species. And we have some significant p-values that we consider significant that come for only 5-6 data points, coming for 2-3 papers. So this is quite limited because it's quite limited to a single paper. Or sometimes we say a lot of things about some aphids that studies all come from the same laboratory. So these are limitations that we mentioned in the paper, which is now under review. And but I wouldn't say it's a limitation. The limitation is the data that it's out there. And as long as when you write a meta-analysis, you are saying, hey, this is what we have. This is what a meta-analysis is about. Meta-analysis is about saying we don't have enough data in this species or in this system, so future researchers can try to fill this gap. For the publication bias and the p-hacking, we tested it in all our models, and we didn't have any evidence for publication bias or p-hacking. I've done another meta-analysis in another project.
Sorry, can you just explain very briefly how this is done if people are interested? Because I guess lots of people do meta-analysis.
So basically, well, that's a tough question.
No,
so
if it's short, if you can do it shortly.
So the p-hacking is very simple. You just draw a curve on your p-values, and usually you have to have few p-values of 0.04, a few more of 0.03, and quite a lot of p-values which are very small. If you have, it's the opposite. Most of the p-values are very small, and only few p-values are close to 0.05. If you have the opposite, it means that you have done an experiment, the p-value was 0.06. Oh, I want a 0.04. You repeat the experiment, you repeat until you get the 0.04, and then you publish that. And that's
so it's an excess of p-values close to 0.05.
Yes, so there's more p-values close to just in between 0.04 and 0.05 than there should be. Okay. And the publication bias is when only very significant effects are published. So when you see like the variation in your data in some p-values is larger when there's more. Let me see if I say it well. You have more variation in your effect sizes in the less significant effects. So you have a plot which you have a kind of triangle, and the dots should be inside. It's like a kind of a normal curve.
We already had a question from Alexandra. So Nicolas Sauvion is saying a comment about Bemisia tabaci, which is in fact a complex of species, as you mentioned, and thinks there might be a risk of biasing the results by not treating these species, subspecies separately. So Nicola perhaps can expand on this or react to your. So what do you think?
Nicola cannot talk.
Okay, so well, anyway, then Enrique,
no, I understand the question, and it's a very good question. In fact, we haven't thought about it, and that would be quite simple to include, just as we did for aphids, that we separated between species of aphids. We could separate between Bemisia tabaci biotypes. There's the Mediterranean. There's 3-4 biotypes. If I remember well. Yeah, the problem is that we don't have much data from whiteflies, so even subsetting it into biotypes is going to be complicated. And also, not all studies provide the biotype. Many studies just set Bemisia tabaci. So, one important message about meta-analysis, doing a meta-analysis, is that when you publish a paper, experimental paper, try to think about the person that will someday do a meta-analysis and try to give all information in a clear way, so it saves time.
Yeah, okay, so guess perhaps one last question by Alexandra, since you did all these different orders or taxa, so what about beetles? Where are the symbionts in beetles?
I think Alexander, you need to read our book chapter. I mean, it's not freely available, but just send me an email. I'll send you the whole book if you want. I hope the editors are not in the audience, but I'll send it to you because it's our effort. Even if you are supposed to pay, I'll send it to you for free. So in beetles, okay, let me think. Well, there's a lot of evidences in beetles. There's important research done in the States, also in Germany. Most symbionts in beetles are in the guts, but I think there are some examples of Tortosia beetles which are inside the body. I don't remember exactly, but mostly they are in the guts. And
so you have the information somewhere, otherwise, if you.
Yes. Check. In our review, we did quite some. We cited quite some of these papers, but I don't remember every single. The paper has more than 200 references, so it's a lot of information there. And but they are mostly in the guts, and these in the guts, what the insect eats, then these microbes can assist in digestion. But in beetles, one very important thing that came in our review is that there's not many obligatory symbionts in beetles, because many beetles have acquired genes from beetles and they have incorporated into their own genomes by horizontal gene transfer. So they, instead of keeping the bacteria with them, they have acquired the genes that they were interested and they keep them for themselves. So beetles are one of these cases where we think symbionts are not like always there helping in digestion, because most of the genes were acquired by horizontal gene transfer. This is one of the key points of our review too.
All right, I think we can stop here. So someone was asking about where you can get the video. So do you agree that this video is shared from your talk? Or is it
yes, I think my hair is not looking that bad. Yeah.
Okay, nothing very confidential. So someone can step in and remind people, like Alison or Emmanuel, where can get the video again? Is it from Agrinia? Or is it on the website of the LabX?
The last time we published the video, we were sent the video on Monday that we put on the LabX webpage. But to access it, you need to put the date of last week, of this week, because otherwise it just updates to the most recent date. For example, on Monday, they'll only have it from showing dates from Monday, but you can put the date back to today to see Frago's poster, where we'll put the link.
All right, not sure I understood, but it will be available on the LabX webpage.
Yes, it'll be available on the LabX website, but you'll need to find Frago's poster to access it.
Yeah. Okay.
All right, okay, so thanks again, Eric, and thanks everyone, and I guess we'll see you next week, Friday, same time, unless it's someone from I think it's still normal time next week.
No,
Bye-bye, have a nice weekend.
Friday is at 4.
Oh, sorry. Oh, so next week it's at 4 because it's someone from the US.
Thank you.
Bye bye. Have a nice week.
Bye bye.